I'm with Felipe at Compound Genetics at Las Vegas 2024, MJBiz.
Yeah.
How's show going for you so far?
It's been great. This is amazing to be able to sell seeds here for the first time, and then it was really nice to, we've never had a booth here before, so it was awesome.
Amazing. We went to a grow facility over at Planet 13, and I was talking to your guys about genetic markers and really getting into the nitty-gritty of breeding, and they said you're the guy to talk to. So they said, get into what you're doing in regards to genetic markering, able to identify problems ahead of time. And
so we spent a couple years growing everything out and then phenotyping it. So we took all the phenotypic data, like the agronomic traits, and say, hey, here's the stem width. This is how tall it grows. This is how quick it does. Everything from root size, 165 different properties.
Yeah.
And then we went ahead and actually took a larger probe, sequenced the entire library, and then boiled it down to about 1,000 SNPs and made our own probe where we added 25 markers from other literature that had been published. And so we're able to go ahead and plot where everything lives on a PCA plot and use that to make our selection. So it's dramatically accelerated breeding for us. And if we're looking for a particular target or looking at an outlier, it just shines on the charts.
So you basically take two drastically different plants that would never normally come together in nature and hybridize them.
That's right.
And get something super unique.
Yeah, that's where you get that hybrid vigor from taking the furthest apart from each other.
Yeah, neat.
And so and then aside from that, identifying what loci of interest that we want to target to add to future probes of the SNP. So this SNP, these are the traits we're seeing with that. So it's a long process. Yeah. But the more we do, the more information we get.
Super. So basically, it's going out of the dark ages into finding exactly what is going to be the thing that works.
So we made like a strain comparison tool where I can check the boxes of like 3 or 4 strains and pop up the drawer. And now you can see all the different cannabinoids and terpenoids strain by strain, comparing strain. So as we're going and developing, we take that, not only the genotype, right, from the sequencing, but also the analytical data to help us put all that together. Basically, here's a phylogenetic tree of all the different strains, and this isn't necessarily if they're brother and sister and came down the same lineage, but they're living next to each other, generally where we see same terpene. So one pheno may be over here and it may be closer to another strain. And so they all kind of live on this phylogenetic tree.
How cool is that? Thanks,
cool, man. Cheers.
Good to meet you, man.
Yeah. Nice to meet you.